Let's talk about the criminal in the room, aging. Now, as postdocs, we're only getting a little bit older. But what's interesting is that our brains are really susceptible to aging. And what we often see as researchers, we only see the brain at the end stage, after years and years of genetic and environmental influences. And sometimes these influences are so great that it can actually accelerate aging, speed it up so much that we get a process known as neurodegeneration, which is when the cells of the brain start dying. Now let's look at the brain like a crime scene, right? So here on the slide, I have a picture of a neuron, beautiful cells that are very abundant within the brain. Now, since it's a crime scene, we have to gather some evidence. And researchers have been doing this for years and years. And what they see is so universal to the point that if we look at one protein, TDP43, we can see it misplaced within these cells. It moves from one compartment to another in 97% of all ALS cases, which is one form of neurodegeneration. But if we look at another form, such as dementia, we see the same protein misplaced in over 50% of cases. Now, this is just one protein. There are many others that we also like to look at. But the question becomes, why are these proteins in the wrong place? Why have they moved from one compartment to the other? The work that I do in the lab looks at the transport of these proteins, how these proteins move from one compartment to another within these beautiful cells. And specifically, what we want to do is we want to find that if we can target these transport pathways, can we restore the function of these proteins? Because as these proteins move from one place to the other, not only do they lose their original function, but they also gain another function, which we really don't want. These proteins, they gunk up, they accumulate, and they aggregate. And what can happen is that this by itself can actually propagate pathology, further contributing to this form of accelerated aging, culminating in neurodegeneration. Now, what we see in the lab is that if we target the pathways which move these proteins around within the cell, we can actually reverse this gunking process, right? We can start to see that we see less aggregates, we restore nuclear function, which is the function of these proteins in their original compartments. And hopefully one day, not only can we start to treat these neurodegenerative diseases, but maybe I'll be giving this talk to you as a grad student, because maybe we reversed aging for a little bit. Thank you so much.